we all are familiar with small molecules, and most of the audience might be familiar with docking of small molecules to a given target. But today I would like to introduce you to another class of molecules, which are carbohydrate mimetics. Part of the tradition, I would like to acknowledge the traditional owners of the land where the Queensland University of Technology stands. We pay respect to their elders, lords, customs, and creation spirits. On my right side, you see the, a beautiful view of the campus. And you can see there is a government house on site at the campus. So, Alex, I think next time, you should attend the conference in person. That will give you an opportunity to visit Australia and also visit us at QT.
I would love to.
Before 3 days, before the war started, I think, for the last 2 years, we have heard about the COVID-19 pandemic, right? We all are familiar with COVID-19. For 2 years, most of the countries were in lockdown, especially Australia, which has opened our international borders this week. So the COVID-19 pandemic so far, has caused more than 8.1 million cases and more than 500,000 deaths. As we all know, COVID-19, one of the key proteins that's responsible for the infection is called the severe acute respiratory syndrome coronavirus protein spike. Prior to vaccination, clotting disorders are well recognized in infected patients, which led to severe complications, including hospitalizations, right? And that in case of the Delta strain of COVID-19. It is appealing to understand the molecular mechanisms and address ideas that may involve therapeutic interventions. So we already, we all got vaccinated. Probably, we all got the booster dose. But still, there is plenty of room in future to come up with a therapeutic intervention for targeting COVID-19 and other similar viral families. One of the key proteins that plays a major role in the infection in COVID-19 is spike protein. It is a glycoprotein. The two-dimensional structure of this protein, as you can see in the schematic, it's a massively big protein, right? And this is just the monomer of that. The sequence just shows the monomer version of the protein, right? And it can be divided into two domains, S1 and S2. The S2 domain is partially embedded into the membrane, whereas the S1 domain, is outside the membrane. So it allows interaction with different proteins, including the host membrane, right? In January 2020, there were a lot of crystal structures, EM structures that came out that provided detailed information at atomistic level about the structure of the S1 domain of the spike protein. Remember, it's a glycoprotein. So what you are seeing here on the screen on the left-hand side is just the protein in its naked form. But because it's a glycoprotein, imagine there will be lots and lots of sugar molecules and post-translational modifications attached to it. So that means not the entire surface of the protein is accessible for targeting with a small molecule or an antibody or a peptide. Physiologically, this protein exists as a trimer. So you can see in the schematic on the left, it's a trimer. You have a domain called receptor binding domain, right? receptor binding domain is the primary domain that engages with another protein called ACE2. Now, this ACE2 helps the spike protein to enter the host cells and establish respiratory infection. Now, looking at the, the strial EM structure, you will see a lot of RBD domains. So there are 3 RBD domains in the trimer. 2 of the domains are in the down conformation, and 1 of the domains you can clearly see in green is in the up conformation. And this is the conformation that interacts with the ACE2 protein on the host cell. On the right-hand side, you will see, the green color blob is ACE2, and then the blue color one is the receptor binding domain of spike 1. This is the monomeric representation of the complex. In the red color, you see receptor binding motif that interacts directly with ACE2. Is that this should be the interface that should be targeted if you were to design any therapeutic against COVID-19, right? Now, let me point out to you, we have the N-terminal domain in spike 2, and that's where majority of the neutralizing antibodies, they interact with spike. Last few years, many research groups have tried to figure out the mechanism of action of the spike protein. So here you can see, right? Here you can see the ACE2 protein from the host membrane. This is your COVID-19, right? And you can see the spike trimer here. ACE2 is a key protein that interacts with spike. But besides ACE2, you also need a molecule called heparin sulfate proteoglycan. This heparin sulfate proteoglycans, they are usually found on the extracellular matrix, right? And they are massively, they are several kilodaltons in weight, and they are very flexible. So what happens, as you can see, along with ACE2, these molecules are also available. They drive the conformational change of the spike trimer, right? And then, that's how the host, that's how the spike protein will infect the host cell. Now, if we were to target, any therapeutic against this mechanism, what we need is to block this interaction, right? So what happens when heparin sulfate and ACE2 interacts with spike protein? As I mentioned, it drives the spike trimer from a closed conformation to an open conformation. It also leads to the cleavage of furin site on the spike protein. Now, not to be confused with the word heparin and heparin sulfate proteoglycan. These are 2 distinct molecules. In the previous slide, I was talking about heparin sulfate proteoglycan that acts as a cofactor for spike protein. Now I'm referring to, if you were to target this mechanism, I'm referring to heparin as one of the inhibitor molecules. And in a second, I will come down to the differences between these 2 molecules. As I mentioned, these heparin sulfate proteoglycans are members of extracellular matrix along with other proteins like collagen fiber, fibronectin, and so forth. They play even a very critical role, not only in diseases like cancer, they also play a role in tissue regeneration. A lot of biomaterial scientists working in the field, they try to mimic the binding of these molecules in their polymers, right? Now, in clinic, we use a derivative of heparin sulfate called heparin. So the clinical form that we use, the anti-coagulant that we use in the clinic, is called heparin. If we look at the general structures of this molecule, it's a repeating disaccharide of sugars. One is called the glucuronic acid and the other is called glucosamine. So one is an amine, one is an acid. This sugar have substitutions like sulfate and acetylation group and so forth. And you can see the two sugars are connected by a linkage, right? It's called the glycosidic linkage. So the carbon of one sugar at position 1 connects to the carbon of another sugar at position 4. So it's 1 to 4 linkage, right? And you can also see there are different places where you can put anionic substitutions like sulfates, right? So in case of glucosamine, you have a carboxyl group. And at 2 positions, you can also attach a sulfate group. Whereas in case of glucosamine, you can have substitutions at basically 3, 4, 5, and 6 positions. Now, if we look at the clinical version of heparin, which is called fondaparinux, the pattern is very interesting. If you focus on the third sugar in the figure middle, figure D, you will see that there is a sulfation at the 3 position. So normally you find sulfation at 2 and 6 position. But in this scenario, you have sulfation at this position, which gives us specifically the anti-coagulant activity. Imagine the number of rotatable bonds in this pentasaccharide. Any guesses how many rotatable bonds are there compared to a small molecule? So if you were to model such molecules, you have to deal with high degree of freedom because of, you have the torsions between the 2 sugars, and you also have the torsion when you attach substitutions on the sugar. So a pentasaccharide or a hexasaccharide can have up to 56 rotatable bonds. If we look at the NMR structures of this molecule, so there is way back in 1990s, Barbara Molloy, she came up with the first NMR model of this molecule. This is a dodecasaccharide. That means you have 12 units in this model. And as you can see, just like DNA, it forms a very nice little helical structure, right? But now, besides the degree of freedom, we have one more complexity here. Remember, we are dealing with sugars, right? And now this the acidic part of this disaccharide can undergo conformational changes, and they exist in 3 different forms depending on the substitution of the sulfate group. So here you can see a 4C1 conformation for glucosamine as well as the acidic moiety, glucuronic acid. The other form of this sugar, it can be also iduronic acid, and iduronic acid can exist between 1C4 form or 2SO form. When we refer to 4C1 or 1C4 as a chair conformation, whereas we refer to 2SO conformation as a skew boat conformation, right? And you can see, depending on the ring conformations of the sugar, the orientation of the sulfates in this helical structure can be different. So in figure B, you can clearly see the iduronic acids are in both conformations, whereas in figure C, you can see, the sugars are in skew both conformation. Now, these molecules achieve selectivity or specificity when they bind to the protein, either through their negative charge. So as I said, you can play with sulfation or acetylation. You can play with the length of these molecules. Again, ring conformations play an important role. Not only when, not only in the structures in solution, but also when they are bound to the protein. I showed you the example in previous slide that these molecules can be sulfated at 2 positions or 6 positions. So it can be, you can have sulfation at the NH group, and you can also add sulfation at the O position, so N and O sulfation. As I mentioned, a pentasaccharide or a hexasaccharide can have up to 56 rotatable bonds. And not to forget, I'll show you the examples. These molecules, unlike, small molecules, they don't bind to a well-defined pocket. So we have a lot of challenges while trying to model this molecule. Here comes example of several classes of proteins, and they have different pathological roles. They are involved, all this heparin binding protein or GAG binding protein. They are involved from various mechanisms, right from cancer to wound healing to tissue development to bone remodeling and so forth. And the striking feature here in this figure, they don't bind to the pocket, except one of them enzyme called heparin, which I will be talking later. But if you can see, most of these molecules, they either bind to loop-like region, very shallow pockets. There are very few secondary structures around the binding site. Now, another key factor while designing this type of molecule, we have, like when I was doing my MPhil and PhD at Curtin, we have shown that the GAG binding sites are unconserved across different species. And this is a very key factor that we need to really consider while designing molecules, because ultimately you are going to do animal studies, right? You are taking your therapeutics to animal models, and you need to be really cautious about which animal model is the right model to test such kind of molecules. So before I jump into, the tool that we have developed for docking such molecules, I wanted to introduce to you, certain properties of this molecule, because I will be referring to those properties while analyzing the conformations of this molecule. As I mentioned before, the ring conformation can exist between 431, 134, 2s0 conformation. And this flexibility of the ring is called puckering. This is a very common phenomenon even in RNA and DNA. So, so you see this happening in 5-member sugars or 6-member sugars. Here is an example in figure B. So you know that that's a typical disaccharide scaffold of such molecule. Then in figure B, the colors highlight the key groups that are important for binding to antithrombin, that's responsible for the anticoagulant activity of this particular pentasaccharide, right? Now, one would imagine that, hey, one would argue, Neha, you have lots of sulfates and carboxylate here. One should keep in mind, yes, ionic interactions are important when it comes to sulfates or carboxylates interacting with base residues like lysines and arginine. But it's also important, the hydrophobicity also plays a very important role. So in case of the crystal structure of antithrombin and heparin pentasaccharide, they have shown hydrophobicity dominates over anionic interaction. Okay, now this is how would we represent this ring puckering? So to a layman, we just look at the theta. or phi in this case, okay? So we just look at the angle 0 degrees, 90 degrees, and 180 degrees. And if your angle is at 90 degrees, then I'll show you in, few slides down the line, I will show you how you can further differentiate, narrow down the conformations of the ring of the heparin. Figure E shows how do we calculate the torsions between the two sugars, right? So if we look, just like proteins have phi and psi angles, this molecule also have a nomenclature for phi and psi angle. How amazing is that? So you can see in the blue the atoms that are defined as phi, and the angles in yellow or orange, you can see they are defined as psi. Okay? And so how do we address all these issues, or the conformational properties of this molecule, and especially when they are bound to the protein? Now, one way of doing this, so let's draw some methods from, somewhere from 19th. When people started looking at high throughput crystallography, they came upon a method called solvent crystal structures, right? That means you throw different fragments in along with your solvent. When you're trying to crystallize the protein, you try to throw as many fragments as you want in, and they get crystallized along with the protein. And it's assumed that that's the most likely binding site for a given fragment or a given solute molecule, right? So I have a movie here. Let's see if it plays. So here I have thrown 8 disaccharides. You can see the protein is a receptor binding domain of SARS-CoV2 spike protein, right? And you can clearly see the conformational change. In the protein, you can see that tiny little helix going conformational change. The loops undergoing conformational change upon binding of the fragment. This is one way of looking at or trying to find the binding sites for the sugars. But of course, now another issue is, what do you do with very long, massive molecules? Because of course, this molecule do not exist only as a disaccharide, right? They are several kilodaltons in weight. Therefore, we have looked at a docking method. It's published. This work was done by a student, Eric Bottier. He was an honor student and he did an amazing job in collaboration with Professor Vito Ferro. If Vito is here in the audience, not sure. This method is pretty much very much based on, Ringer. Orthodox Ringer is a very commonly used package for performing virtual screening of small molecules. Rob Wood's group at here, they try to look at Ringer carb for docking of other sugars. And now we have tried to optimize the same package Ringer for docking of glycosaminoglycans. This includes even other types of glycosaminoglycans, which I will not be discussing today. I will be focusing mainly on heparin and heparin sulfate in this talk. Now, as you can see, just like every docking program, you first prepare your protein, you define the box if you know the binding site, either through experiment or your co-solvent molecular dynamic simulation, or either by, other programs that predict the binding site. So you define a box around the protein, and then you run docking, right? And it uses the same algorithm. All we have tried to do is try to parameterize the torsion angles using DFT methods. Okay? So people think, yes, these molecules are highly flexible. But if we look at the crystal structures of these proteins or the NMR co-crystallize with the sugars or the enema structures of heparin and heparan sulfate. They are flexible, but they are within a range. So the phi and phi angle, they are within a range. So can we apply a penalty while docking if these angles go outside a particular range? Yes, absolutely. We can apply that penalty. Now the other issue, most of the time the crystal structures with heparin or heparan sulfate, there is an eye effect because you have four crystals with more than one monomer. So, you have an eye effect from the neighboring molecule. Furthermore, these crystal structures are not very high resolution structures. There are issues with the refinement of the structure. So initially we started refining the structures. Initially we do 86 PDB structures from the protein data bank that were co-crystallized with this ligand. However, towards the end of this refinement exercise, we narrowed down to only 12 proteins in the data set. That's one of the biggest limitations. Now, another limitation, if you look at the orientation of the side chain that interact with the carboxylate, all the sulfate groups of this molecule. So we have to use DFT methods again to look at, how well this DFT methods perform to reproduce the interaction, the salt bridges between the protein and the ligand. And we were quite actually pleased with the outcomes. You can see when we try to redock and generate the crystallographic pose from docking over 10 trials, you see the alignment is pretty good. Now, let's not forget another issue with sugars. You have reducing and non-reducing ends. So in docking, your molecule is very likely they enslaved the other way around, and the RMSD may not be able to capture that. So to capture that RMSD, we use another method called sucrose. Here is the performance of glycosaminoglycans. Be mindful, this training set only includes 12 high-resolution crystal structures after refinement from PDB redo. They are not directly from protein data bank. So compared to, a famous docking program, Glide from Schrödinger, then we have the original Winger version here. We have the Autodoc 3 version, which was, in early 90s and early 2000, Barbara Mulloy and her group, or Rebecca Wade's group, they were very much used Autodoc 4 for docking of such molecules. So compared to all commercially available programs and some of the open source programs, Glycosaminoglycans did an excellent job when we compare the RMSDs in reproducing the crystallographic pose. And now I will move on to talking about the applications of Glycosaminoglycans. So interest was to decipher the binding site of the receptor binding domain of called SARS-CoV2 spike protein. And we initially use a Plus Pro version for docking. So Plus Pro, they have a provision for docking of a heparin tetrasaccharide, and it's a standard tetrasaccharide. You cannot play with the sulfation pattern, you cannot play with the length, but it gives you fairly a coarse-grained mapping of where the likely binding sites are on the surface of the protein. On the left, you can see the electrostatic potential surface of the receptor binding domain, and you can clearly see the population are centered around two major binding sites. You can see one site running across, and then the other side on the right, slightly towards the right in the loopy region, which I call as site 2. So you can see the entire surface in blue, which will act as site 1, and then another site as site 2. If we rotate the molecule further, you will see another site which we address as site 3. Now, let me remind you, this site 3 is the receptor binding motif. And, most majority of the efforts to design small molecules or peptides have gone into targeting site 3 because it directly targets the ACE2 interface. Now, we carried out, docking of longer oligosaccharides and shorter oligosaccharides, starting from tetrasaccharide all the way to octasaccharide. Now, if you look here, after taking into account the conformational changes, you can see a longer oligosaccharide can be accommodated on site 1 of the RBD domain, and you can clearly see the conformational changes highlighted in yellow upon binding of the ligand. On my right in figure B, you can already see that a shorter oligosaccharide, can be accommodated on site 3. And let's not forget, this is the site where ACE2 binds directly with spike protein. So in previous slide, we were just looking at, the monomeric version of the receptor binding domain. But in reality, spike is a trimer, right? What happens? Consider the trimer. So we carried out post-solvent molecular dynamic simulations on the trimer. This is a massive system, more than 180,000 atoms. So you can see all the green blobs, they are heparin disaccharide, whereas you can see the other moieties in blob formation. In the sphere representation, they are the glycosylation site. Remember, I mentioned to you, spike is a glycoprotein. So not the entire surface is not available for the ligands to bind, right? Because this glycosylation will inhibit binding. They will not allow the ligands to bind. They will hinder the surface, right? This accessible surface area will be much lower in presence of those glycosylation sites, right? So we have done some collaborative work with Professor Vito Ferro and Professor Jeremy Ernbull in the UK, and DSS study, differential scanning fluorometry, which also looks like, how the protein folds or unfolds at a given temperature, And you can clearly see, so we have heparin with different sizes, and you can clearly see that DP16, which means if you have an oligosaccharide of length like a 16-mer, it clearly binds the spike protein compared to just having a water as a control. Now, you must be wondering what is UFH? UFH, unfractionated heparin, which means the version that you use in clinic, the fondaparinum, is a pentasaccharide. They are for small molecular weight heparin. Unfractionated heparin can be several kilodalton. Now that's ambiguous. It can be a heparin sulfate. That means not necessarily everything has sulfate. It can also have acetylated groups on it. It can be a mixture of number of fragments, 32 mers, 42 mers, or a 12 mer, or even a disaccharide. So they are massive in molecular weight. Now, based on our results, we identified even heparin binding sites in the N-terminal domain of the spike protein. So remember, when we looked at the monomer, we were only looking at the receptor binding domain. Now, in this scenario, we are looking at the N-terminal domain. As I mentioned before, the N-terminal domain forms the binding sites for several antibodies. Now, in this example, you can see the orientation of the ligand. This is heparin sulfate. Remember, heparin sulfate acts as a co-receptor, not as an inhibitor. And you can see not all residues are sulfated. So you have sulfation towards the end region, towards the reducing and non-reducing end, but in middle you have non-sulfated region. But if we try to model heparin, which is a fully sulfated oligosaccharide, right? Look at the orientation of this molecule. Strikingly different. You cannot see, clearly it's not bringing different domain. And you can see it's orienting differently. And it also represents, if you can see, so this was a 12-mer. That clearly shows that if you were to have a longer oligosaccharide, or polysaccharide that runs from receptor binding domain all the way to the furin cleavage site or to the end terminal, then you need a 35-mer or a 40-mer, right? So far, this way heparin. Heparin is an inhibitor. So a lot of other groups, such as Michael Boone's group, have shown heparin is an inhibitor for spike. Here we have a mimetic called pixantimod or PC545. It was synthesized in group of Professor V. Ferro, who is also a prominent user of salt with CISR. As you can see at the structure of this molecule, it has an heterosaccharide followed by a lipophilic tail. It's not cholesterol, it's cholesterol-like, it's cholesterol moiety. You can see the electrostatic potential surface. On my right, you see the hydrophobic surface. The blue indicates positively charged region surface. The red indicates negatively charged surface. On my right, the blue indicates hydrophilic and the orange indicates hydrophobic. And you can clearly see the tetrasaccharide sits nicely on the blue region, making electrostatic interactions with charged residues, right? And towards the top, where the ACE2 receptor binding site is, you can see the cholesterol or the hydrophobic moiety interacting with tyrosines and phenylalanines. Remember, there are tyrosines, and in different viral strains, this tyrosine have been mutated. There are mutations around this region in alpha, beta, delta, and omicron strain. I want to highlight here, this is the original Wuhan strain of the virus, but in the other variants, most of the mutations have turned into a basic residue, making it even more basic sites for heparan sulfate to interact with. Now, if we rotate this molecule, so in this one here, we are referring to site 1, right, as an active site for this molecule. If I rotate the molecule, there is also a possibility of a second binding site for this molecule. Look at the surface one more time. The tetrasaccharide sits nicely at site 3 of heparin, where the heparin was binding. And you see the cholesterol moiety going towards the other side, towards the hydrophobic pocket, right? So in collaboration with the group in the UK, we have carried out, again, DSF studies as well as circular dichroism, which is a very common method used to look at the conformational changes of the protein. You can clearly see the behavior of this mimetic, heparin mimetic. It's very different compared to heparin and the native, only the RBD domain. It's in presence of, PC545 or pixantimod, this protein or this particular domain undergoes a conformational change. And to heparin. And so I wanted to mention that pixanthimol was originally targeted at heparinase. This molecule was developed in Queensland. It has an anti-cancer activity as well. It's currently undergoing phase 2 trials for solid tumors, as well as is undergoing phase 1 trial for COVID-19 treatment in Brazil. Enzyme. It's not a glycoprotein. And as you can see, highlighted in figure A on my left, you can see 2 distinct pockets for binding of heparin. Now, not to be confused between heparin and heparin sulfate. In case of COVID-19, heparin sulfate was a cofactor, and heparin is an inhibitor. In case of heparinase, here fondaparinux, which is the clinical version of heparin, is a substrate. Okay, so heparinase will cleave fondaparinux at this sugar. Can you see? Between residue E and S. Okay. Now, as I mentioned earlier, that most of the time, this molecule, they bind on the surface of the protein. But in this scenario, the substrate is bound according to the crystal structures by Gordon Davis from the UK. Again, this crystal structure shows a well-defined tetrasaccharide bound very deep inside the cavity, right? Now, remember, PH505 or pixanthimol is an inhibitor of heparin. It's not a substrate. Here we have used biosolvate CSAR version 10.2 to figure out docking of fondaparinux. So far, the crystallization of force with inhibitors, with heparinase, they have not been successful. We only have the crystal structures of heparinase in presence of substrate, like heparin tetrasaccharide, or the analogs of that molecule, or there is a small substrate with a probe attached to it. As I mentioned, the clinical version fondaparinux is a substrate docking, taking into account the smaller substrate as a reference, and CSAR did an excellent job at docking of fondaparinux. You can see clearly the superimposition of the ring with the co-crystallized structure. The green one is the smaller substrate, which was co-crystallized, whereas you can see the dock results, the fondaparinux dock onto it. We were quite happy with this. But now, when we try to, dock the ligand pixanthimol in this pocket, even by including or extending the binding site on the either ends of, heparin binding site, HBD1 and HBD2, we had no luck trying to dock this molecule. Not surprising, right? Look at this pocket. The pocket here, you can see it's a mix of hydrophobicity and hydrophilicity, okay? So again, reiterating my, coming from the first slide, I would like to reiterate, it's not only the ionic interactions that play an important role. Even hydrophobicity plays an important role when it comes to modeling of such molecules. But before even I jump into docking of this mimetic, heparin mimetic, let's worry about the solution structure of this mimetic, okay? So we have recently published, like in January, and in fact today, Rita sent me this paper where, now it's online and the volume number has been assigned to this manuscript. We try to understand the conformation of this molecule in solution. So we perform MD simulations of this molecule. Remember, It's not straightforward. Although we have charm and glycan force field to take into account the conformation of the tetrasaccharide, but here we have to mix and match the force field. So you have a cholesterol-like moiety attached to it. So the tetrasaccharide was modeled using the glycan force field, whereas we modeled the cholesterol using the lipid 14 force field from the Amber 20 package. Okay. Repeat it 3 times. Its simulation was for 500 nanoseconds. And you can clearly see. So let's count the lipophilic group as residue 1, and then moving from there onwards, I will count residue 2, 3, 4, and 5. If you can clearly see, the MD simulation is sampling the conformational space quite well. Now the sugars attached to this lipophilic tail, they are glucose, and clearly you can see from figure B, C, and D, this figure indicates the glycosidic torsion between each residue, so the phi and psi angles. Remember, the phi and psi angles are not representative of what's happening to the ring conformation. So one can see the torsions have converged very nicely, but what's happening with the puckering? Now we are looking at the ring, and you can clearly see on the right-hand side, especially the residues that are attached to this lipophilic tail, the first sugar that are attached to this cholesterol moiety, it undergoes ring puckering. And it fluctuates. So you can see our theta is between 180 to 160, and then it jumps between 80 to 90 degrees. And when I showed you in previous slide, the figure 4 puckering, you can narrow down when it's theta is 90 degrees, you can sub-classify the ring conformation. And you can clearly see 3S1 conformation, OS2 conformation, and 3OB conformation. So this, in general, this has skew both conformation. And when you have theta equal to 180, you have, chair conformations. This was for residue 1 of that tetrasaccharide. But what happens to the conformation of the other 3 sugars? And they all are in 4C1 conformation. Now let's look at, docking using glycotorch and then running MD simulations on the dock courses. So from docking, we got 2 prominent clusters. One where the lipophilic tail, is pointing towards heparin binding site 2, as you can see in figure A. And if we do relative free energy calculations using a method like MMHBSA, which is not an accurate method, which has its own assumptions, but still it helps you to, relatively rank the binding poses. If we look at pose A, the cholesterol is pointing down, and remember, there is a hydrophobic pocket in that region as well. Now, PH545 doesn't exactly bind deep in the pocket. Imagine you have a vessel, okay? You have the substrate binding into the vessel, whereas, PH545 or this mimetic, it acts as the lead of the vessel. Okay, so it binds on the surface, it doesn't go down deep in the pocket. And then the second point in figure B, you can see the cholesterol is pointing towards HBD1, towards the upward side. Now, pose B is very interesting because in the MD simulation, that cholesterol tail is flopping around. Now, that creates, room for a substrate to enter the pocket. Now, as you can see, we tried docking, of the substrate in presence of pose B. So you have PH545 sitting there where cholesterol is pointing up. And if you try to dock that substrate, it will easily dock. So that means smaller substrate can bind heparin in presence of PH545. or other way around, P545 can bind, in presence of the smaller substrates. But if you have a molecule like fondaparinus, which is a pentasaccharide, then it's not possible because fondaparinus is going to occupy the entire pocket from HBD1 to HBD2 of the smaller substrate. But if you have something longer than the disaccharide, for example, fondaparinus is a pentasaccharide, you need, it cannot compete for binding with P545. And clearly, to summarize, Glycotorch outperforms a range of commercial and open source programs when docking large GAGs. Glycotorch also takes into account the ring conformation while docking. Remember, we have tried to parameterize the torsion angles as well in the scoring function. Talking about the applications of this program, we have shown heparin sulfate binds to the SARS-CoV2 spike 1 protein, especially the receptor binding domain, the furin cleavage site, and the N-terminal domain. So, docking as well as co-solvent MD simulation plays a key role in trying to predict the binding sites of such molecules. We have also shown using experimental data and modeling, the protein undergoes conformational change upon binding to heparin or is mimetic. Clearly, you can also investigate mimetics of heparin for binding to SARS-CoV2 spike protein. As I mentioned, before, compared to the Wuhan strain, the Delta, Alpha, Beta, Gamma, and Omicron have undergone several mutations. Now remember, this mutation, you should not base your judgment just on the mutation from the RBD domain. In this variant, the mutations are also present in other regions of the spike protein, including the furin cleavage site, including the N-terminal domain. Now, all these mutations have converted, the hydrophobic residues or the non-charged residues into basic amino acids, which suggests that these basic amino acids are capable of interacting very strongly through ionic interactions with heparan sulfate as a cofactor. Today only, we got an email, we have finished all the experimental work using P545 and other assay techniques. This was done in the UK. The manuscript is currently under review in ACS Central Science. Watch out for it. It will be published hopefully in a month's time. And finally, I would like to summarize that we have successfully, tried to correlate the conformation of this heparin mimetic P545, with the NMR observables. And this molecule, the mimetic, binds to heparin, is capable of binding to heparin in the presence of smaller substrates in the pocket. And with that, I would like to acknowledge, of course, my students. They form a great team, and my collaborators, without whom, this work on either on COVID-19 or, in general, the GAG field wouldn't have been possible. Professor Diedrich, whom she introduced me to this field during my master's, during my MPhil. Professor Winter Farrow, we have successfully published many papers over the last 3 to 4 years. And I very much value his collaboration, the chemistry side of things. Professor Jeremy Turnbull from Liverpool, who's led us all the DSF, CD, SPR, and IEC studies. This work wouldn't have been possible without the support of high-performance computing resources like NCI and QHPC, and without funding from Advanced Queensland and Center for Genomics and Personalized Health, and of course, Q&A. With that, I would like to welcome questions from the audience.